Assalamu alaikum. Good evening, everyone. It is my honor today to moderate a session, actually a very important session to speak about the role of SARS-2 virus rapid antigen as a POCT testing during the pandemic, how we can contain or support containing the pandemic using the rapid antigen tests. My name is Ashraf Dada. I am the section head of the immunovirology and molecular virology sections at King Faisal Hospital in Jeddah. The elected president of the Arabian Society for Histocompatibility and Immunogenomics. And I will start and moderate this session. I will start with a topic which is, I think it is important during the pandemic to show the role of the laboratory, the critical role actually of the laboratory in containing the pandemic, and particularly in details about the clinical implication of the rapid antigen in detecting of SARS-2 virus. I just share my screen. I hope you can see it. So the title of the presentation is a clinical implementation of SARS-2 rapid antigen POCT as a test which help and support the clinician and the hospital in containing the COVID-19 pandemic. In this presentation, I will mainly focus on 3 points. The number one is in general, the critical role of hospital-based laboratories in containing the COVID pandemic. And the second one is the important role of rapid antigen detection tests in containing the pandemic. And at the end, I will introduce avenues and the future approaches of using the rapid antigen detecting test in containing this pandemic, but also further pandemic. Now we come to the first point, which is the critical role of hospital-based labs. As I mentioned, actually the lab is a central facility in each hospital, and therefore play a critical role in a hospital. This is the main objectives of the lab. First of all, of course, to generate accurate clinical lab results, and this is the normal situation. However, preferable that the lab participate actively in innovative and adequate diagnostic options, in the development also of such options, and introducing of new technologies in the hospital. This is the role of the lab. And ideal if the lab also have ideas to practicable interpretation of the results, of the accurate results was generated. It's not enough to generate just results. If you want to have an ideal situation in your hospital, especially during a pandemic situation, the generated results should be interpreted correctly and match the hospital need, as I will show you also in this presentation. For these accurate results, participation in the development and in the introducing of new lab technologies and correct interpretation of the generated results. You need, of course, knowledge. I mean with knowledge, technical knowledge, but also know-how behind this knowledge, expertise, and in general, all issues behind technical testing. And of course, quality control, which an essential point in each actually testing. This is also needed during a pandemic situation. And the research as well, because research during the pandemic opened new avenues. open new windows and new ways for you to better contain the pandemic. I think with these three columns, every lab can participate and play a critical and important role to contain the pandemic and help the hospital in managing the pandemic, because pandemic, as you know, always a very difficult situation and a very challenging situation for each hospital. Starting, for example, for the gold standard PCR, which should every laboratory actually be able to do PCR test. However, even PCR test is very challenging when you want to do it correctly and interpret it correctly. The results. What mean these results for you? It is really positive, or it is false positive, or maybe true negative, or what does mean this results? It should be corrected. If you have correct positive results, for example, CT value 35 or the 40, what does mean this CT value of 40, 45 even? What all this mean for your interpretation? What you should do when you have persistent situation in your laboratory? You have always positive PCR results. What you should do in this? If you haven't this knowledge and you haven't maybe the science also and research, you will not be able to interpret these results correctly, and thus maybe you will not help your hospital optimally as it should be. As a control, for example, I show you example from our hospital, our lab, hospital-based lab, how we were able to support our hospital and established accurate diagnostic laboratory with, as I mentioned, innovative procedures, innovative technologies, and correct interpretations of the results. At the beginning of our testing, immediately we have compared the new results, compared our results with external facility. For example, in our case, we compared our results with the results of the National Health Lab in Riyadh, Saudi Arabia, and we found a concordance. This gives us, of course, more confidence in our work and show us that we correctly work. Not only this, we compared also our positive results with the viral culture results. Is this positive, for example, and CT value of 20 or 25? What does it mean? It means really that the virus existing, or just maybe residual RNA. And we, as you can see here, hundreds of specimens were tested and compared using S3 laboratory. And this gives us more light into the life of the virus, of the SARS-CoV virus. We know that, for example, according to our published data, we know that CT value above 20 most likely means inactive virus, and CT value below 20 most likely mean active virus, and the patient is contagious. These things and experiment are needed in order, as I mentioned, to correctly and ideally interpret your results correctly. As you can see, we have many publications about, for example, this COVID-19 testing, including viral culture results and other and CT values, of course, what does mean. And this make the workflow in our hospital much easily. This is, for example, based on our published scientific data, which we adjust the interpretation of the PCR results. For example, at our lab, from the beginning, we have made this experiment, and we found that, for example, CT value above 35 mean that viral culture negative, and the virus is avital, is not alive. So, and therefore we interpreted this results as a negative. If you put such results in your lab, for example, as a positive results, it means a lot. The patient must be isolated, and a lot of headache for your hospital. This is what I mean with knowledge and scientific experience to be able to correctly interpret your results during a pandemic. This is also another example which also, with this knowledge which we had a donor, a kidney donor who was positive, PCR positive. But as I mentioned, we have investigated what our CT values mean. And this donor had a high CT value, which according to our results and viral culture results, which we published, it means that the virus is vital. It means that this donor is not able to infect the recipient of the kidney. So we were able to transplant 2 kidneys from a donor with positive COVID-19 with high CT value. And I have written a manuscript in preparation. The manuscript is already published. Also, what we have done at our laboratory, we started to collect antibodies from recovered donors and transfuse this antibody or the plasma of these donors to patients with COVID-19, mainly in the ICU, mainly critical ill patients. Also, this clinical trials are done by the laboratory, and with this way also we support the hospital in containing the pandemic. Also, we discovered that the COVID or the SARS-2 viruses is able to trigger the building of HLA antibody, class 1 and class 2 antibody. As you can see here, strong HLA antibody. This also were diagnosed at our laboratory and give us, for example, information that these patients who has already recovered from SARS-2 viruses, maybe they have HLA antibody, and we should consider this if these patients will be transplanted in the future or they will get, for example, a transfusion. In these cases, the HLA antibody could be relevant. All these things, what you see, could you make in your laboratory to support the pandemic. Also, a simple actually test the antibody testing in your lab for the SARS-2 antibody testing, serology testing. With this serology testing, you can know that, for example, the vaccination status of your employee in your hospital. When they get, for example, vaccine, they develop enough SARS-2 antibodies or not. But also for the patients, when you have a patient, for example, difficult patient who is not recovered, you can see if he has enough antibody or not. If not, you can also adjust plasma therapy and give him passively antibody from recovered donor. That's what we done also at our hospital. Another thing also which the lab can do is the tracking of variants. It's also a rule and a critical rule of the laboratory. We established also at our lab in King Faisal Hospital in Jeddah the next generation sequencing to track the variants, to know, is this variant what we have, this positive results what we have, is, for example, Omicron or Delta or something else. We have now new variants, as you know, BA1, BA2, BA4, and BA5 as well. So, and with this way, you know the kind of variants which circulating in your hospital. For example, when you look for this figure, and this is from December, we had the most of the cases was BA1, as you can see here. 1 month later, only in February, you can see here that the number of BA2 increased and compared to January. Also, we established our lab as a reference lab for NGS, SARS-2 NGS sequencing by the GIS database, international database. Also, what we have done from our laboratory, we make thought about difficult patients who should be vaccinated. Which time they should vaccinate? For example, in this case, patients with hematological oncological disorders. Which time they should receive the vaccine? And if they receive the vaccine, did they develop antibodies against SARS-2 virus or not. And also we publish this data. This is in general what I saw the critical role of a hospital-based lab in general in containing the malady. It's not, as I mentioned, just generate results, but also participate in developmental labs and in research as well. Now we come to the important role of rapid antigen detection test. As you know, the rapid antigen detections has increased dramatically in the last weeks and months in the diagnosis of COVID-19. Because it has a lot of advantage. You don't need S2 laboratory, for example, with all this complicated and complex things which you need to install. Just you can do it as a point of care in a normal room and under normal air conditions. You don't want all this extraction and all these things what you need, all this to wear, all this protection clothes, all this is not needed. You can save all these steps, also the amplification and all this huge number of stuff and all this. You can save all this critical also steps in your laboratory by doing this rapid antigen testing. Now, how do rapid antigen testing work? They work most of them, not all of them. The most of them working according to the lateral flow test work. How does the principle is quickly, as you can see here, almost all rapid tests. They have here an antibody which called the primary antibody. This is mobile antibody. It's movable antibody. So, and in the second line, you have a fixed immobilized antibody, and in the third field you have a secondary antibody. And when you have a protein of the virus in the, for example, nasal or the nasopharyngeal swab, you put it in the camera, in the chamber, and if the result is positive, the protein of the virus will be catched by this antibody and also by this antibody, by both the primary and the secondary antibody. So in this case, you will have a positive results in the tests, in the specimen itself, and in the control as well. So in this case, it's positive. But if you don't have the virus in your specimen, so there's no protein antibody or the antigen antibody reaction will happen, and this mobile antibody, which is not fixed in the chamber, will move, as you can see here, to the fixed antibody and catch by this antibody, and you have in this case only positive case in your control, but negative case in the specimen line. And then you can interpret, of course, your results. All these technologies at the beginning, there were a lot of difficulties to introduce and produce such tests. I remember the first test which we validated at our hospital, it was zero specificity and zero sensitivity. Now we have a high quality tests, antigen tests with high specificity and high sensitivity. This is, for example, this test which we use at our hospital, among other tests. As you can see here, this is the content. I will not go in details. I think it will be explained by the further presentation. Anyway, a critical role which I mentioned, which lead often to false negative results, is a critical role to how to take the specimen from the patients or healthcare workers, for example, to do it correctly from the nasal or the nasopharyngeal and according to the manufacturer instruction. It's a very critical point in releasing the results and will mixing the specimen in the reagents as instructed by the company. This is very critical. We observe also here wrong results when these steps are not done correctly. And now the antigen tests, as I mentioned, are increased dramatically because this is actually like the PCR. When you look to this graphics, the PCR works immediately after infection, as you can see here. When the virus infection happened, you can diagnose also the virus. Same applied also for the rapid antigen tests. You can also very early detect the viruses, which advantage, and detected mainly at the first stage of infections. So based in this, because this test is very quickly and now with relatively high specificity and high sensitivity, we evaluated and make a study at our hospital to de-isolate vaccinated COVID-19 healthcare workers using antigen rapid tests. Previously, the patients were isolated, or the healthcare workers were isolated based on PCR results, which is the gold standard. Now we tested if we can isolate the healthcare workers based on the results of antigen testing. This is a single center study was done at King Faisal Hospital in Jeddah. This is a retrospective cohort study for 1 month from 11 January to 12 February, which is all healthcare workers with positive COVID PCR results. In this study, we included only healthcare workers with mild symptoms. Mild symptoms need headache, a little bit fever, nasal congestion, runny nose, sneezing, or sore throat. The most was actually upper respiratory infection and not lower respiratory infections. And this is the results and demographics data. The most of the tested healthcare workers were female, 63%, which means 303 out of 480, and male were 177. And the age of the tested healthcare workers, as you can see, the vast majority was below 35, and from 35 to 44 was 38%, and above 44 is 21%. So the most of them actually relatively young people. And as you can see here, we have about 16% physicians and 40% nurses and 46% other employees. And this is very important information on this slide. The vast majority of these healthcare workers were vaccinated with the second dose, 75%, and 25% was third dose at the time of testing with the rapid antigen tests. And the average duration between the last vaccine dose and the breakthrough infections was 52, because I mentioned that all of them was vaccinated. Despite this, they become a breakthrough infections. And this information also important between the infection and the vaccination. It was 52 days. It means relatively fresh, as not very old vaccination. Despite this, we have breakthrough infection because of all ongoing variants of SARS-2 virus. And this is the positive and negative results at day 7 of post symptoms. Day 7 after onset of the symptoms, they were tested with the rapid antigen tests in this study. And we found that 36% of these healthcare workers still turned positive. And however, 64% were negative and could be released and come back to work based on rapid antigen test at day 7, as I mentioned. And this is what we observed from January to now April, that the positivity rate by rapid antigen test declined continuously, as we can see here. We don't know exactly the reason. The reason is, according to our study, is not the third vaccine or third dose or second dose, but most likely are the variant, because the variant, as you know, is different. In January, we have the BA1 variant dominated, then BA2, now we have BA4 and BA5 variants, as you know. So, and these variants have less diagnostic window, but also less days of symptoms. And therefore, maybe the healthcare workers were released earlier, and the majority of them were released based on the antigen rapid test. This shows also the important role of the antigen rapid test in the isolating patients in a hospital-based laboratory. This is what I mentioned. You can see here no difference between second dose and third dose in terms of positivity. At day 7, we found the healthcare workers with a second dose, 35% were positive. With the third dose, even more, but not significantly more. As you can see here, P is 0.239. This is at all not significant difference, but it means that is independent of the second or third dose, the positive rate is not influenced by the vaccine. And therefore, I mentioned most likely are the variants are the reason for this development and different. As I mentioned, these results are also comparable with finding of other studies, which shows a decline in the positivity rate of rapid antigen detection test among 26 healthcare workers was tested in this study from 46 to 38, similar like our results in day 5 and day 7 of post-symptom onset. Now we come to the last point of this presentation, which is the avenue and the points which the important role for the rapid antigen detection test, which we need in the future, quickly and fast. As you can see in this slide, this, for example, from our hospital also, we face also, like in the kingdom, 4 waves. Actually, now it's the small wave here, a very small one compared with this huge wave. With this huge wave, we started here to use the rapid antigen tests. Without using the rapid antigen, you will be overwhelmed. You will not be able to manage a quick spreading pandemic. And this actually shows us the important but the critical role of rapid antigen tests in containing a fast spreading of a pandemic. And we wish that such rapid antigen tests will be developed earlier in a pandemic. In this case, they are developed in the corona case a little bit late. Not, for example, here in this, during the first wave, we haven't rapid antigen tests, or at least not sufficient, like is the case now, which we can use it without big difficulties also to manage a huge wave, as you can see here in this slide. So it means a symptom-based test approach using a rapid antigen is a useful tool in the de-isolation of health with mild disease in the area of Omicron. Now, as I mentioned, we have BA4 and BA5, and you know both this are most likely spreading faster than the BA1. So it means that we need the rapid antigen test in these for these variants more urgently even than the BA1. And this also demonstrates the role of the rapid antigen in the future. Also, the rapid antigen detection tests should be used not only by new variants, but also by severe cases. As I mentioned, we have tested only healthcare workers with mild symptoms, but also we need here to test it in what is about severe cases with which are in the ICU. Maybe when we have, for example, more accurate rapid antigen testing with a sensitivity and specificity 100%, for example, then we can use it also for ICU cases, not only for health care workers. But also we need antigen testing for further another pandemic. For example, all of you know about the monkeypox disease, which now is spreading. We have now about 27 countries. As you can see, there's updated statistic data from the WHO, and using the antigen testing, which should be probably more challenging than the SARS-2 viruses, will help and support us in containing this infectious disease and control it from fast spreading. However, as I mentioned, we need to have challenging we need to rapid antigen from the cross and from the vesicles of affected patients, and we need here also a higher sensitivity rate than it is the case now. In conclusion, fighting a pandemic needs a rapid response and a quick response and the use of all lab resources effectively. Besides routine diagnostic, which is not enough in fighting a pandemic, you need innovative research, advanced rapid technologies like that, for example, for better management and a quick control of the pandemic. At the end, I would like to thank all these people behind all these works from the molecular lab and HLA lab as well, and the blood bank and all involved colleagues. And without their support, we will not, of course, be able to do all these tests which I introduced for you. Thank you very much for your attention. And I received the information that we will continue with the further sessions. And at the end, you have the opportunity of all the sessions to ask questions. Thank you very much. And I will now continue to introduce the next speakers. The next speakers it is my honor actually to introduce the next speakers, which we work together in the lab. Ms. Elaf Al-Amoudi, she is a medical technologist at King Faisal Hospital in Jeddah. She is also acting POCT coordinator and ASCP certified. She is also AACC POCT specialist program certified, and she actually the person behind establishing the antigen rapid SARS-2 viruses at our hospital. Please, Elaf, the floor is yours.
Thank you very much, Dr. Dada, for the introduction. Let me share my slides. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone. It is Elaf Al-Amoudi. I'm a medical technologist. Currently, I'm acting as a POCT coordinator at King Faisal Specialist Hospital and Research Center, Jeddah branch. Today, I'll be presenting COVID-19 rapid antigen test. In this presentation, we'll go through overview of virus, SARS-CoV-2 structure, laboratory PCR testing, what does CT value mean, testing process for the rapid antigen test, results interpretation, the principle of the test, validation process for COVID-19 antigen rapid test kits. Overview of the virus structure. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2. Virus is enveloped, caused by coronaviruses, which is belonged of a family Coronaviridae. The virus is a single-stranded RNA virus. Coronavirus are composed of four main structural proteins, as you can see in the figure. We have spike protein, envelope protein, membrane, and nucleocapsid. Laboratory PCR testing or polymerase chain reaction, as we all agreed that it is the golden method and it has the highest sensitivity and specificity to detect COVID-19 viruses. The PCR assay positive reaction is detected by accumulation of a fluorescent signal. The CT or cycle threshold is defined as the number of cycles required for the fluorescent signal to cross the threshold. So what does CT mean? CT levels are inversely proportional to the amount of target nucleic acid in the sample. The lower the CT level, the greater the amount of target nucleic acid in the sample. So how is it relevant, CT and COVID-19 virus? So there is various cycling number depending on the type of an instrument. Based on the literature review, the low cycle number will have high copies of virus. So if you have a high viral load, you'll get low CT value. And if you have low viral load in the sample, you'll have a high CT value. Sorry for that. So beside PCR testing, we have a COVID antigen test that can be used to detect coronavirus. And CT actually helped us to understand more the sensitivity of the antigen. For example, if CT is 25 or below, then the sensitivity of the antigen test will be more than 80% based off different evaluation studies that we've been conducted and we've been read about it. On the other hand, if the CT value greater than 25, then the sensitivity will be subjected. If the patient is symptomatic and antigen tests tend to be negative, it is highly recommended to confirm it by alternative method PCR. There is several routes available commercially with the variant type of sensitivity and specificity. And this is very important for the lab to do the validation to find the best assay benefits of the SARS-CoV-2 rapid antigen test. And we have been validated several kits from different companies. And in the next slides, I'm going to show you the one of the kits that we have been validated. And this is the kit SARS-CoV-2 rapid antigen test. The following components are needed for test and included in the kit. As you can see in the figure, we have the test device, we have extraction buffer tube, we have nozzle cup, and sterile swab. The test process for the SARS-CoV rapid antigen test, as you can see below, our swab was nasopharyngeal. So the steps as following. You insert the sterile swab until you reach the nasopharyngeal cavity. You rotate the swab 3 to 4 times. You insert the swab after that in the buffer tube, mix it 5 times, remove the swab. While you're removing the swab, squeeze the buffer tube, especially on the cotton bud, to remove the remaining liquid from the cotton to the liquid. Close the buffer tube, apply 3 drops on the cassette well. Read the results after 15 minutes, but not more than 30 minutes. We don't read before 15 minutes to avoid any false negative results. Results interpretation. So results are qualitative. It's either positive or negative results. Each cassette we have internal quality control line. So the control line should appear whether the result is positive and negative, and it will assure us the validity of the cassette. So for the positive results, you will have line in C line and line in T test line. So if you have both lines, the results are positive. No matter how strong or fine the line under the test line, once it's appearing, the test is considered positive. If you have only one line under C, control line, the test is negative. And if you don't have a line under C and you have it under T, or if you don't have any line at all, neither under C or T, the cassette is invalid and you need to replace it with a new one. A benefit of the SARS-CoV-2 rapid antigen test, getting, of course, a quick results within 15 to 30 minutes, easy handling, no instrument required. Test principle, the SARS-CoV-2 rapid antigen test is a rapid chromatographic immunoassay The results qualitative, either positive or negative. It detects SARS-CoV-2 antigen, the nucleoprotein. This point is a very good point because most of virus mutation, like we had alpha, delta, omicron, all mutation happened in the spike protein, but they are all shared the same nucleocapsid, which leads to this test to be able to detect any kind of COVID-19 virus. Next slide is our data for the validation of the COVID antigen test. We choose this company. The company claim was their specificity is 100%, and their sensitivity is more than 95% if CT value PCR equals or less than 30. So we did two studies. One we compared the antigen test with the golden method PCR, but all samples were with CT value 30 or less, and we did the other study regardless what the CT value. So for the first study, we had 18 nasopharyngeal samples. 6 samples were negative by all test methods, PCR and antigen. 12 samples were PCR positive and CT with CT value equals or less than 30. 1 sample only was positive by PCR and negative by antigen test. 11 samples were positive by PCR and by Roche antigen test. None of the samples were negative by PCR and positive by Roche. This is a valid point, proves that the specificity of the company. Even though the number that we did is very small, after we implement the service in our hospital, we did ongoing validation. We compared all PCRs with the antigen and for 2 months, and we still have a very good specificity. This is the data in detail. As you can see in the table, we have 11 true positive, and we have 0 false positive. We have 1 false negative and 6 true negative. So the sensitivity is 92, which is excellent, good. It met the manufacturer claim, and it is meeting our accreditation and hospital policy criteria to validate any qualitative assay that we have to have 80% or more. So 92%, it's a good percentage. Specificity 100%. Discordant antigen negative PCR positive. CT value PCR equals or less than 30, only 1 sample. Agreement antigen positive PCR positive CT value of PCR equals or less than 30, 11 samples. This is the second study based on PCR testing results regardless of the CT value. Sorry. Total number are 22 nasopharyngeal. 6 samples were negative by all test method, PCR and antigen. 16 samples were PCR positive. 5 samples were positive by PCR and negative by antigen test. 11 samples were positive by PCR and by antigen test. None of the samples were negative by PCR and positive by Roche. And here again, the test is proving its specificity. The results, the data in detail, as you can see in the table, we have 11 true positive, 0 false positive, 5 false negative, and 6 true negative. Here, the specificity is still 100%. Sensitivity is less than 80 because of the discordant that we had antigen negative PCR positive 5 samples. But when we looked at the results of CT value of those 5 samples, all of them were PCR CT more than 30, which is expected, as the manufacturer stated in their manufacturer package insert that their sensitivity is high when the CT value is less than 30 or less than 30. Agreement, antigen positive and PCR positive, 11 samples. To conclude, actually this test as a point of care rapid antigen test helped us a lot during the pandemic. And it is a very useful tool to use. It is helped us in expediting the admission, the clearance, and even decreasing the waiting time for the patient. I just want to mention that each health organization should have their own criteria, flow chart, or algorithm to follow in ordering the test or reporting results. And the physician or healthcare givers should bear in mind and consider the symptoms beside the test antigen. So if we have symptomatic patients with antigen negative, it is highly recommended, even by CDC, to confirm it with alternative method or PCR. That's all for my time. Thank you very much for listening. We'll go back to Dr. Data. Be safe and shukran.
So thank you very much, Ella, for this really excellent presentation and impressive data, actually. And as I mentioned, we will have the opportunity later to discuss the results and the presentation. Now we come to our third presentation this today session. It is my honor to introduce Ms. Rani Bseso. Rani Bseso is the Medical Affairs Manager for Roche Diagnostic in Saudi Arabia. Rani has extensive experience in pharmaceutical and diagnostic industry, and he is working in this field since more than 20 years, and he is handling the COVID-19 issue from medical aspects and point of view. And I think we have organized other big events together, and it is really my honor to hear you, to see and hear you today. Rani, please, the floor is yours.
Thank you, Dr. Ashraf. Good evening. It's my honor to be with you here in this successful event. I will go directly and share my presentation. So I have just a very simple presentation, but very important in content, which is when should we use the rapid antigen test? So from medical point of view, the best utilization for the antigen point of care testing will play an important role to use it in terms of medical and clinical value to maximize the efficiency. And therefore, we can share with you 4 important cases that can be used during or for point of care testing antigen. The first case is triage of symptomatic patients, testing for asymptomatic individual after non-exposure, complementary to lab testing, and the 4th one is more convenient and less invasive sampling. We will go in details for each case in order to know the rationale behind each different scenario. So as we can see here, do I need to isolate myself? This is very important and common question. And we'll go to the left-hand side of the slide, which is point of care rapid test enable faster decision making on self-isolation, reducing the risk of further spreading, which we all look for. So the purpose of this case is to have fast triaging and decision making for patient management for symptomatic patients in particular. And the benefit out of this is symptomatic patient as well as everyone in their surrounding will be benefiting out of this, using the point of care testing. And the sites where we can do this in hospitals, clinics, physician offices, test hubs, public locations, occupational medicine, pharmacies, and other. And the Coveted is triage or more or less diagnosis. Second case, the scenario is saying, I have been in close contact with an infected person. Have I caught the virus? This is also very important and practical question. I'm sure all of us during the last 2 years, I would say, we have asked ourselves this question. And let's go again on the left-hand side. The result from SARS CoV rapid antigen test nasal can help healthcare professionals to prevent further spread if an individual has been exposed to the virus, or reassure those showing a negative result. So what is the purpose of this case? So the purpose for timely reaction when higher risk groups or sites have been exposed to the virus. So therefore, who is going to benefit out of this scenario? Individuals and their contacts following exposure to an infected person. Sites, primary care, clinics and physicians' offices, test hubs and other tests. And the caveat should only be applied where there is a real risk of exposure and an increased probability of infection. Case number 3. How do we follow the advice to test, and test if we simply do not have the capabilities? And we go again, point of care testing, rapid antigen can fill the gap if lab capabilities are unable to meet exceptionally high demand. They also facilitate testing when travel to at least location if not possible. So the purpose of this scenario of providing antigen point of care testing is to provide access to testing for everyone in need, regardless of infrastructure and mobility. And who will benefit out of this? Symptomatic patient and individual with known exposure, like high-risk groups and those in contact with these people. For example, healthcare systems, organization, and governments. Where we can implement this in hospitals, clinics, physician offices, test hubs, public locations, occupational medicine, pharmacies, and other. And the caveat, not intended for testing of asymptomatic patient without known exposure, in order to give the value for the test and the efficiency of the use. Last but not least, the 4 cases, and the question is there a less invasive method for testing of more sensitive patient? And therefore, the nasal sampling method simplifies the testing procedure, causes less discomfort for patient, and offers more protection for healthcare workers. So the purpose behind the scenario is to provide an alternative testing method for more sensitive patient and improve protection for healthcare professionals. Who will benefit out of this case? Test operators, less complex handling, steps, and better protection as described by the previous speaker. Patients also reduce patient discomfort. Healthcare providers like faster procedure. Where we can implement this scenario, the traditional and non-traditional testing location, like public locations, schools, and daycare, nursing homes. The caveat, test performance can be lower compared with nasopharyngeal test. So before I close the presentation, I would like just to summarize. There are very important 4 cases for using the point of care antigen testing, and those are triaging of symptomatic patient, testing of asymptomatic individual after known exposure, complementary to lab testing, and the 4th one is more convenient and less invasive sampling. And by this, I finish my presentation, and I would like to thank you for your active listening, and I will give the mic for the next speaker.
Thank you, Rani, for this presentation. As I mentioned, the questions at the end. Now we come to the next presentation. The presenter is Mr. Anas Al-Fayoumi. Anas is working also at Roche Diagnostics, Saudi Arabia, and he is the responsible marketing product manager for POCT testing, and it's an important task also for the SARS-2 antigen rapid test. Please, the floor is yours, Anas.
Thank you, Dr. I would like to thank all of the attendees to attend this very important webinar. My name is Anas Al-Fayoumi. I am the point of care product manager in Roche Diagnostics. I will share now my slides. Okay, can you see my slides? Just somebody confirm.
We can see it.
Okay, so today I will present Roche SARS Coveted 2 rapid antigen point of care test portfolio on Roche that's testing in your hand. So testing in your hand, the SARS CoV-2 antigen self-test nasal provides reliable results for patients suspected of having COVID-19. The self-test can be performed by general public, which provides another option for decentralized testing whenever and wherever needed can be the basis for making safer decisions to protect yourself and others. So here, the self-testing help fight the pandemic. Let me to say what Dr. Tedros, the World Health Organization, had said in the first beginning of this pandemic, that you cannot fight a fire blindfolded, and we cannot stop the pandemic if we don't know who's infected. We have a simple message for all countries: test, test, and test. And Europe posed rapid COVID-19 test to control outbreak early in 2021, and protocol on implementing the self-testing forwarded to secondary school today, which was on April 2021. Approval of at-home test release a powerful pandemic fighting weapon. So testing everywhere at any time, self-testing offers a holistic solution where individuals collect a specimen, perform the test, and interpret the results by themselves. No medical assistance is required, while the test can be performed everywhere. Here, let me just to give you the key benefit of the self-testing, that we can keep it in the room temperature and no instruments needed. The usual shelf life will be about 24 months after the production for safer security. And we have here sensitivity and specificity. The sensitivity talks about the ability of a test to correctly identify patients with a disease, while the specificity, the ability of a test to correctly identify people without the disease. So we have here sensitivity 91.1% and specificity 99.6%. So the result will be after 15 minutes, and it's nasal sample. Here is the each kit contains this contains 5 tests here, and we have a buffer tube holder, and we have 5 sterile swabs, and we have the extraction buffer tubes, the nozzle cap, and the instruction for use, and the quick reference guide, and of course always available in Arabic since it's for patient self-testing. Here I will not go through the construction of the virus since Ilaf had mentioned about what's the construction of this virus, but let me here to mention about why we selected, or why the companies selected the nucleocapsid antigen, selected based on the experimental data on manufacturability, so performed better than the other target in several hundred clinical samples, and can be synthesized in suitable form, large-scale production in a fast and reliable manner, and main performance requirement for selection. So we have high specificity to the SARS-CoV-2 SARS. We have the nucleocapsid protein and membrane glycoprotein, spike protein, envelope protein, and usually we are targeting the nucleocapsid protein. Here is the test principle. Usually we have here the sample bag contains the SARS-CoV antigen. We have the conjugate bag, which includes the mouse monoclonal anti-SARS-CoV-2 antibody gold conjugate, and we have the purified chicken IgY gold conjugate. And here is the nitrocellulase membrane, which contains the two lines, the test line and the control line, which includes the mouse monoclonal anti-SARS-CoV-2 antibody and the mouse monoclonal anti-chicken IgY. So here, the line of the control must be appears in each test. If no control line is visible, so test result should be considered invalid, and we have to repeat the test again. So here we can talk about the needs of the key users for patients, allows for testing without appointments in uncontrolled zone, governments and societies, fight the pandemic, protect societies and economics, healthcare system saves time and capacity, further supplement to lab testing, and health care professionals increased safety due to less contact with possibility infected patients. So here we can summarize the benefit to the patients, that they can get reliable answers, high test quality and performance, quick results. No need, just within 15 minutes we can get the result. A step towards together for social issues, easy handling without need to see the healthcare professionals to schedule an appointment, peace of mind since you can get a result on the spot, easy and safe handling. Anybody can use this type of test. So taking the test everywhere, in the school, in the home, in the factory, in the company, in the hospital, anywhere in your office, you can make this test a tool to open. So self-testing provides the professional audience with a safer testing option for opening the economy, real-world evidence, independent performance studies with large sample sizes, including professional collection, self-collection, and self-testing groups, and increased, of course, safety, the main objective for us in any society. So here, let me to just go about how can we handle, as our partner in King Faisal Hospital mentioned, Ms. Ilaf, that's here we need to get the nasal swab, and then we will insert the swab into the extraction buffer tube, which and squeeze the tube and still at the swab for 10 times, and then remove the swab while squeezing the side of the tube. Press the nozzle cap tightly onto the tube, then drop the samples. We need 4 drops of samples, and we have to read the result in 15 to 30 minutes only, and we have to be careful. Don't read the result after 30 minutes. So here we have usually in 2 lines, C and T. C for control and T for test. If the patient if the result had been appeared here in the line C and T, even if it was bright, it means that the patient have the positive COVID-19. If here the C line only, had appeared, it means that the patient is negative. And as I mentioned before, the C line must mandatory to appear. If the C line doesn't appear, it means that something is wrong, and we have to repeat the test again. Let me now to move to the second type of antigen test in Roche. We have here, let me to summarize about why we need the need of quick or developmental antigen test at point of care. So here, the clinicians and patients face various testing challenges that highlight the need for a quick and trust antigen test at the point of care. So in the environment, the lab testing is not always available. Result feedback waiting times can disrupt necessary actions, and organizing screening of groups of exposed individuals is difficult. So in these three cases mainly, we can use the antigen rapid test, nasopharyngeal one. I am talking here about the nasopharyngeal, here about differences between the lab testing. So in the lab testing, we need to make a patient consultation, take a swab sample, send the samples to the lab, wait for lab results, arrange follow-up with the patient and patient consultation. While here in the point of care environment or decentralized location, so here we just need the patient consultation, test in the office, in the home, and anywhere. Test in the office only, in the office of inside the hospitals, and then the result it will be within 15 to 30 minutes. So it's more comfortable for patients, less time and effort for the healthcare professionals. Here is the content of the any healthcare nasopharyngeal antigen test. So we have the test device, we have instruction for use, we have also the quick reference guide, we have the sterile swab, nozzle cap, and extraction buffer tube, similar like others. So here we can identify the nasopharyngeal testing as a rapid chromatographic immunoassay for qualitative detection of specific SARS-CoV-2 antigens present in the human nasopharynx, intended for use on individuals suspected of COVID-19, result within 15 minutes, and readout window from 15 to 30 minutes. Here is the this study had been done by Roche Diagnostics, actually, about the sensitivity of this instrument. For this test, no instrument needed, as I mentioned. So the performance data shows here the sensitivity and the specificity for the rapid antigen test from a pool of 220 positive testing patients with a CT value less than 30. So the rapid test reported only 10 false negative results, meaning 95.5% sensitivity of this test, and for the specificity from a pool of 727 negative testing patients, the rapid test reported only 6 was false positive result, meaning that the specificity is 99.2%. Here, I will move now for the third type of antigen for healthcare professional use also. Here, let me just to highlight what's the difference between the nasopharyngeal and nasal testing. That nasopharyngeal swabs are the sampling gold standard, delivering high performance of reliable results, while the nasal swab, as you can see here, this is nasal and here is nasopharyngeal. That's why we choose to, for the self-testing by the patient, by the public, it will be a nasal one. So the nasal swabs mean less invasive testing, providing a great alternative for more sensitive patients, let us to say. So in this environment, we can use the nasal swab. Same here. We can keep it in the room temperature, and we have two equality control included in the kit. We have a specificity 99.1% and sensitivity 89.6%, and result after 15 minutes same, and it will be self-collection possible. It means here that the patient in presence of the healthcare professional can take the swab, or the healthcare, the nurse can take the swab for the patient, but here must be healthcare professional available near of the patient. Here is the what the kits have involved. Here we have a buffer tube break, we have a sterile swab, we have a test device, we have extraction buffer tube, we have a nozzle cap, we have a quick reference guide, and we have an instruction for use, and we have positive and negative controls integrated with the same. I had finished my presentation. I hope that I didn't get a long time, and this testing in your hand for yourself, and please keep safe. Thank you all for you.
Thank you, Mr. Anas, for this very nice presentation and introducing both nasopharyngeal and nasal antigen testing. And now the session is open for discussion, and you have the opportunity to raise questions. You can write your question in the comment. I hope that you can see the chat and you can write the question there. I don't know if they have the possibility also to ask directly questions verbally or not. If not, just please write your questions. In the meantime, I have a question, if you allow me also, to Mr. Rani. And the question is, do you think that we have some difficulties when we want to have very quick results, for example? 15 minutes, maybe in some occasions, some events are long. For example, when you have area without waiting time, and you have a lot of people who want to be tested, where they should wait for 15 minutes. So this is a little bit limitation factor. Do you think you can work in this test to be more faster than it is the case now, or is it not possible?
You are on mute. Ron, you are on mute.
Thank you so much, Dr. Ashraf, for this question. Actually, as you know, Roche is always having a huge investment in research and development. And the moment that we achieve to have the rapid test in 15 minutes is, I would say, is a great achievement. Considering reducing time, I am sure it is also on the radar of the research and development. Meanwhile, I would say 15 minutes comparing to the gold standard PCR of having sometimes during the curfew, it was taking almost 24 hours to in certain period, in certain situations up to 48 hours. So it's a really very uncomparable time from 15 minutes to 24 hours. So, but I can assure you, Roche is always looking for the perfection to provide state-of-art instrument in providing support healthcare professionals.
No, I asked the question because just when you have area, for example, 10, 20 people want to be tested quickly. So not all hospitals have waiting room for all these patients, and they must wait 15 minutes. This limitation factor. This was I raised the question. And regarding the sensitivity and also maybe the quickness, I don't know if you heard about the nanobody. There's nanobody is a small antibody which when for example, from camel or llama, and they are bind to the protein of the virus more strongly than this human antibody or the antibody from chicken, which Anas introduced. Maybe this will be more sensitive, increase the sensitivity, but also the accelerate the time, to have positive results if this antibody will be used. But I don't know why they are not. Maybe it's difficult to be extracted or isolated. Do you have idea about this?
Yes, it has been touched before in one of our global event, and the answer was, having the antibodies is not, as easy technique. So they have went through multiple steps and factors to find out which antibodies was most practical to be tested and captured by the antibody testing. So as we have to find the antigen or the antibody that suits the chemical reaction or the testing performance in order to achieve the results. But currently the human one is the most suitable, or the available option is the most suitable one for practice. I hope I answered your question.
Yes, thank you. I know this because we are working in a project together with Coast, and we use the nano antibody, and they bind very, very strong to the SARS-2 viruses, even to the small parts of the virus, and go deeply structure of the virus. Do we have any questions from the audience? If not, I have another one. Do you think about use which we have demonstrated and show the critical important role of rapid antigen testing in helping and containing the pandemic? Believe me, worldwide, without these antigen testing, the world will be totally different. Huge difficulties, particularly with the Omicron wave. So this helped us a lot, despite the fact that they are not ideal, of course, but they help and support a lot in the daily business and week. Now we have a winter will coming and spring, and we will have another viruses. Do you have other technique to detect, for example, flu A and flu B with antigen test? We have, of course, the PCR, but using the antigen test, or even maybe monkeypox. We have now, or even the other, infection disease.
Yeah, thank you, Dr. Ashraf. Actually, for monkeypox, yes, we have Roche now through Tibmol, which is already acquired by Roche, providing 3 different type of kits to test the monkeypox. One is general for the orthopox family, one for the orthopox family and toward the monkeypox itself. So it's more selective, and third type as well. So we have 3 different types of kits, and those are all PCR testing. So those are already available.
Antigen rapid test. Do we have antigen rapid test?
No, it is PCR testing.
We have, sorry, Rani, for interrupt. Can you hear me?
Yes.
Yes. We are in the process for registration now. The rapid antigen test can measure the COVID-19, influenza A, influenza B, but now still until now, it's launched globally, and we are waiting for registration now. And we are in the process in registration this type of test. It will include the parameter COVID-19, influenza A, influenza B. So it's in post registration, yeah.
Yeah, I was talking about the monkeypox. Yes.
Yes, thank you.
There is one question I can see, if you allow me, Dr. Ashraf.
Yes, please.
They are asking if there's available in the market, in the shops or the pharmacy. I think this is for
This is for, I mean, yes, you can, for self-testing, you can reach for online. I can't here say the name of any customer or dealing with them, but you can reach at least for online in Saudi Arabia for Noon, for example. Yes. If you allow me, Dr. Ashraf, also, that's a lot of people asked about the CME hours. So, on behalf of SPOCTA, that's as usual for all people attendees that they have to fill in the survey which they will receive at 9 o'clock today. And after filling the survey, they can get the CME hours from clinical chemists or sorry, by the SPOCTA or Saudi Society for Clinical Chemistry. So I am seeing in the chat a lot of questions regarding the CME hours. So that's the procedure usually happened.
Yes, thank you, Anas, for explanation. We have also a question: how can we handle the swab waste, and how long we can collect the waste before discard? Thank you. Maybe Elaf, you can answer the question.
Yes, sure. We will deal with it as we are dealing with any tissue. It will be in a biohazard container, and it will be labeled. And here we can discard the swab and the tools of the kit.
Okay.
The biohazard. Yeah, but then when we want to discard it, we seal the container. It will not be full. It will be full until the 2/3. But then place it in a red bag and contact hazardous material manager for pickup.
Okay, thank you, Elaf, for this question, answering the question. Other question saying why we should wait 15 minutes.
This is for me.
Yeah, or Anas, or
Okay.
product manager. Anas, you are unmute. Unmute yourself, please.
Sorry, yeah. Actually, yes, Doctor. Yes, actually, as my colleague Rani mentioned, that's 15 minutes comparing with the PCR tests that the patient will get the result after 24 or more than 24 hours. It's comparing with this. I think the time is good. We can, for screening, for example, or in a hospital, we can use a lot of nurses, for example, to do multiple tests, since no instrument needed for this swab. So it will be just taking the swab. And since we have, if we have, of course, nasopharyngeal is the golden standard for antigen rapid test. We have also the nasal one, which can be used by the healthcare or under the supervision of the healthcare professionals. So I do recommend to do this nasal test for the patient, since they can use it under the supervision of the nurse. So to accelerate if we have a lot of cases in one time, for example. Until now, we have and similar like all of the companies, the same 15 minutes is available now. So I hope that answers your question.
Yeah, I would like also, Dr. Ashraf, if you allow me to add to what Anas mentioned, is also the 15 minutes required because, we are doing like, if you allow me to say, like a reaction. So we put the solution into the, let's say, a chromatography-based strip, and we would like to wait the liquid to pass through this plate, and this will not happen in seconds. So we would like to ensure that the liquid has passed through the strip in a very efficient manner, and they found out the 15 minutes was the most suitable time to ensure that the liquid passed over the test and the control to ensure getting a good result.
Excellent. Now another question. I have noticed that there were a plate for serial dilution in the last slide. To answer the question, I am right. We are going to dilute the swab in the last kit? That's the question.
The swab, you need just to the buffer itself, it has the liquid, so just you need to how can I say it, put the swab in the buffer, okay, and rotate it for 4 times, and then you can make a drop via the buffer. Yes.
But no dilution, as I...
Yeah. No dilution, yes.
Okay.
That's what you need to do.
No dilution.
Very simple, yeah. Follow four steps to do the result, yeah. And it's now very known by the...
Okay, there's other...
Yeah.
Yes. There's a question: if a patient is infected before with COVID, can she be infected again? Yes, we can see here, yes, particularly, especially when we have a new variant. And other question, do we need a second and third dose? Yes, to have enough antibody and be enough protected, you need a second and third dose and even fourth dose for specific population. For example, people with more than 50 years old and older than 50 years. These are, I think, there is no further questions. We will close if there's no any further questions at this session, and want to thank all of you for this really good event, education and awareness event, I consider it. And I would like really to thank the Saudi Society for Clinical Chemistry, Ms. Maha, for the organization, and of course Roche for organizing also and supporting such awareness events which contribute to containing this pandemic. This is a global responsibility of such a big company like Roche. So thank you, all of you, and wish all of you a nice evening, and goodbye.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye.